When I think of the different times of year, fall is my favorite. And one of the reasons is you've got the beautiful, beautiful colors of all the leaves that are on the trees. Sometimes you just ask yourself, how come? How come they were green in the spring, they were green in the summer, and now you got reds and yellows and purples, blues and all kinds of things? One of the things we're going to be focusing on today in today's class of biology is a certain pigment called anthocyanins. Antho refers to flower, cyanin is blue. We get this pigment from purple cabbage juice. And you can see in the test tubes that I have in front of me that I have purple cabbage juice in each one of these test tubes. They all have about the same color. And how do you get this stuff? Well, it's really simple. Go to your local grocery store, get some purple cabbage, take it home, boil it, and after a few minutes of boiling, all of a sudden the water turns this pretty color. Water is a substance that's neutral. It's got a pH of about 7. There's other chemicals that are out there that are not neutral. The ones that are less than 7, we call those acids. The ones that are greater than 7, we call those bases. They can also be called antacids or alkaline substances. So we've got this range, it's called a pH scale, and it goes from 1 to 14. Right in the middle is neutral, that's 7. What I want to show you today is what happens when we put some of these chemicals in that are not neutral. We already know what neutral looks like. This is going to be our control right here. We can measure everything against the color of this test tube. How nice is that? When the fall comes and the leaves begin to change, there's something that takes place biochemically within the leaf itself. It becomes maybe a little acid or maybe a little basic. In fact, during the same period of time, if you remember from what our illustration was associated with leaves, you could think of your hand as a leaf, you could think of this little stalk that comes off the leaf as the petiole, and then we have this thing called the abscission layer. The abscission layer is the thing that actually cuts off the supply of water and nutrients and chlorophyll from the leaf. So if this leaf was originally green, now when we cut off the supply of chlorophyll to it, now the green begins to fade away and any underlying pigment, let's say it's anthocyanin, will now be expressed. But if that pH is going to be an acid versus a base, we get a different color. And the fun thing about today's lab is here we're just looking at one pigment, anthocyanin. Another one's chlorophyll, another one's carotenoids. That's going to be like an orange, yellow color. You see that with squash and with pumpkins and with carrots, of course. But this is just anthocyanins. And what I want to show you is what happens when we add different chemicals to this solution and we see the pretty colors. So with all that said, I'm going to get on my safety glasses. We're dealing with some chemicals here that aren't so nice. Our eyeballs are very, very precious and we don't want to do anything that would compromise that. The very first chemical that I'm going to take, this smells like a cleaner, and the reason is, it is. This is actually toilet bowl cleaner. And certain toilet bowl cleaners have a certain substance in it called hydrochloric acid. That's HCl, if you take chemistry class. That's a strong acid, pH of maybe 1 to 2. Remember, neutral is 7, and now we're really, really low in the pH scale. What's going to happen when I put that into this solution? I already mentioned, it's going to change the color. We're going to start dripping this in there. And what you'll find is it may not be immediate. Well, we'll stir it around a little bit too as we go. Going to put some more in. Stir it around. And now you're going to start seeing that we've got a reddish color to it. When we look at the pH scale and we're looking at this indicator, an indicator is something, a chemical, that actually will change color based on the presence of an acid or a base, we see that we have produced a reddish color to it. Kind of pretty. Now, there's another acid that we're going to use, and this one is not going to be as strong. It's an acid that we find in vinegar. Vinegar is something that we cook with, something we ingest. And obviously anything that we take into our body and it's okay, we would think of that as a weak acid. This one has a pH of about 5. And the actual acid that makes up vinegar, it's only about 5% of it is this acid, but it's called acetic acid. And we're going to do that with test tube number 2. So we'll just start putting it in there. And one thing nice about having a little pipette like this, we can use the pipette also as a little stirring rod. So we'll stir that around to start with. And what you'll notice is, yes, we are changing the color. Remember, our control is test tube number 3. That's the color that we started with all 6 test tubes. Notice too that I'm putting more in there to try to get a color change. Reason is not as strong. That's kind of cool. A lot of times if we're thinking about the characteristic associated with an acid also is if we do taste it, because it's in our food, it has a sour taste. A good example of something like that would be citric acid. If you think of your citrus fruit, think of like a really sour grapefruit. Egads, that's sour. When we think of acids, we think of a sour taste. Let's move to the other side of neutral. These were under 7 right here. Acids. A comes before B if you think of the pH scale. One of the bases that's well known, we use it all the time, it is called baking soda. That's sodium bicarbonate. The sodium bicarbonate is actually made by our pancreas, and if we think of the acid that is in our stomach, that acid is very, very important. It's there to help to digest our food chemically. That's one of its functions. Another purpose, of course, is that when we're eating food, we may have some bacteria on there, some germs. We do cook things, but you also eat raw things all the time. And so that acid bath that these pathogens, these microbes, these bad guys fall into will now get killed just because the pH there is very, very, very strong. It's about a pH of 1 to 2. Found in our stomach. But there's a problem with that acid. As soon as it gets into our small intestine, which is the next part of our GI tract, we're going to find out that acid would cause some of the enzymes that are necessary to digest the food get turned off. They get deactivated, which is just the opposite of what you want. You want digestion so you can absorb it into your bloodstream and then get the nutrients to your cells so that you're able to get energy from your food. But if we didn't have sodium bicarbonate available produced by our pancreas, we would have an issue right there. Here's another chemical, you've heard of this one before, it's called ammonia. Now ammonia is something that we may use around the house as a cleaning supply. Many cleaning supplies actually are bases. What's a property of a base? They feel slippery when wet. Think of soap. Soap is a base. It's alkaline. And if you were to taste a base, I've never had this experience, but I've heard stories about kids saying bad words and their mom has them suck on a bar of soap. Guess what? It's going to have a very bitter taste. Bases are bitter versus acids are sour. So take a look at this color, and we should have a green color. The very last one that we want to show you is going to be a very strong base. Just to give you an idea, if our neutral is about 7 for water, baking soda is going to be about 8, ammonia is maybe 9 to 10, now we get into something called lye, and lye is greater than 10. Remember, 14 is like the worst. But this is a pretty good base in terms of it's very, very strong. Very, very strong. If you were to get this stuff on your hands, you could actually feel some chemical burn. A lot of times if you're looking at that pH scale, if you're high on the scale or low on the scale, so a strong acid or a strong base, then you're talking about actually a chemical burn. And now we look at this one, and we get a real pretty yellow color. What's the bottom line here? Is we're looking at these different chemicals that go into this exact same pigment, the anthocyanin. We see that colors can be associated with acid-base characteristics. And if we look at that from the standpoint of leaves on a tree, all of a sudden you can see just the marvel of the fall. You look up there and here's a splash of color. There's a splash of color. Everywhere you look, you see beauty. And if we only had just this one pigment in the leaf, based on the pH, you can get this beautiful, beautiful array, this rainbow of colors, which again is just candy for the eyes.